Hi everyone, this is Kumar. Belongs to Pharmaceutics Department, SNS College of Pharmacy and Health Sciences. We are going to discuss about the antibiotics topics. So, what is the new things in antibiotics at the present trend? That is nothing but novelties in assessing new antibiotics. So, what is the need of assessing new antibiotics? We are in the critical position. There is no advanced antibiotics to treat such a sepsis technology. For example, if the particular patient has built up or it's affected by the sepsis, we need a new antibiotics with the effective combined therapy. So at present, the antibiotics what we are using is the almost going to be resistant for all of the top class pathogens. So in order to avoid such an incident, in order to avoid the patient those who are suffering from sepsis or anything, we need to find a new antibiotics with the help of the cutting edge technologies. That is nothing but what are the new technologies and new pathways are available for antibiotics design. The first is we have to shift in screening methodologies. We need to shift the conventional screening to the new methodological screening. So what are they? First, we need to switch over the phenotypic screening. So you know the meaning of the phenotypic. It's a wild type. So without knowing the target, it is very impossible to develop the new thing. So we know about the particular strain, exactly where they are starting and what is their phenotypic area. Next, target-based screens especially focus on the specific bacterial enzymes receptors by using pharmacogenomics or the genomic sequences area. So pharmacogenomics we are going to use the human set, and genomic sequences tools we are going to use the strain, that is the microbial area. So exactly here we are need the target based screens, means exactly under the particular gene sequence which is responsible for production of the antimicrobial resistance. That target we have to check it and we have to collect the particular gene sequence, and it is going to multiply in the DNA sequencing methodologies or RT-PCR or whatever may be. Finally, we can encounter the particular sequence which is responsible for the antibiotic resistance. Next, hybrid screens, target based with the phenotype, with the combination of the phenotype and the target based with the help of the top class bioinformatics tools. Nowadays it is available. So here we can go for the identification purpose as well as the optimize and the therapeutic efficacy. Next, very important is drug repurposing and repositioning. So repurpose is nothing but existing drugs for the new antibacterial uses. So what is the repurpose? Almost for example, if you take the rifampicin, it's an old antibiotic. Nowadays it's useful for some of the strains. We can control the new strains with the help of the rifampicin. So under the category we can use the conventional antibiotic to control the newly developed strains. Then we are going for the another area, high throughput library screen for the synergistic uses. You know the synergism is nothing but a combined effect. Whether the existing drug along with the new antibiotic will produce a synergism, thereby it achieves the complete microbial control or not. This is one of the indirect methodology by using the repurposing and repositioning of the already existed drug. Next, as usual, AI tools predict the new antibiotic routes from the genomic data. From the genomic data, from the genomic group, there are so many AIs are available. With the help of the sophisticated AI or concerned AI, we are going to identify the new antibiotic roles from the genomic group. Next, targeting novel pathways and mechanisms. It comes under the category of the chemistry and the pharmacology. So it acts as drug targeting, quorum sensing, virulence, biofilms, and dormant states. Here, dormant state, biofilm, virulence, they are the most critical and very dangerous to control. So if you are targeting the virulence producing gene, biofilm producing gene, and the dormant state producing gene, it is so useful to achieve the assessing of new antibiotics. Next, what are the alternative therapies currently scientists they are doing in the intensive research. The first thing is watch therapy or phage therapy. Next, antimicrobial, peptide design and ribosomal switching case. The final thing is advanced tools and the collaborative platforms. What are the advanced tools nowadays we are using in the industry area, especially research and development under the category of the industry. So, AI predicts resistance, it accelerates the antibiotic lead assessment. So, with the help of the AI, we can reduce the time and we can achieve the easy assessing of new antibiotics. Next, crowdsourced initiatives for the compound testing, for example, Corbex with the help of the GoAd. The final thing is natural product rediscovery from unculturable bacteria via genomics. Though it's considered as a conventional area, nowadays it matters. So what are the important matters? By assessing the new antibiotics, by discovering the new antibiotics, and what are the important things for the society. So we may overcome the resistance and we can find the novel and non-traditional antibiotics faster because of the rapid microbiological evolution that present the previous decades when compared to the 80s, 90s, the previous 10 years, the microbiology world, they are find out that microbes are rapidly mutating and they become the more antimicrobial resistant. So here we need to identify the novel and non-traditional antibiotics faster. Next, shorten development timelines, especially how to treat infections. It's very important. So what are the things very hard to treat the patient? Here we are going to overcome by assessing the new antibiotics. Next, it promotes sustainable, resistant, resilient antibiotic peptides. This is what exactly we need under the heading. So we need to promote the sustainable antibiotic which is eco-friendly. Next, resistance and resilient, which is highly comes under the category of the resistance and resilient towards the microbes. Thank you.